Hi everyone, and welcome to Chapter 3 on the Molecules of Life. So essentially what we're going to be talking about today is what's called macromolecules. And now a molecule is just, remember what we learned last week in Chapter 2, is just, again, it's going to be a combination of things like oxygen and carbon and hydrogen and all those little elements that actually make up a larger little molecule, right? So one would just be a element. When you put a few together, that's an actual molecule. So that's what we're going to be talking about today, but we're going to be talking about macromolecules, which means large molecules. So macro means big, and again, molecule means many different little elements and stuff. So that's what we're going to be talking about, and what composes up us and every other living thing on this planet is these larger macromolecules. So let's go ahead and start talking about those. Now, the first thing I want to talk about is what's called an organic compound. And now it's considered organic if it has carbon as a base. Now, this is really like organic chemistry stuff, so we are not going to get super into this. We're not going to get super into the detail behind this graph right here, but I do want you to kind of know the background of it. So anything that is an organic compound is going to be something that is based in carbon. If you've ever heard that carbon-based life forms, so everything on Earth is a carbon-based life form, that's us, right? And every other living thing on the planet. And that's because it is based in carbon, meaning it has a carbon kind of backbone or skeleton to the molecule that makes up all of our stuff. So you can see everything in green right here, these are all going to be the carbon-based backbones, right? The carbon kind of like structure that everything else is kind of built off of. So that's what you can think of when you think of an organic compound, something with carbon. If it does not have carbon, it is not technically organic, right? And so I know you're thinking, you're like, wait, organic, doesn't that mean like grown without pesticides and stuff like that? Yes, that's a term that we typically use organic for, but really when we're talking about chemistry, especially organic chemistry, this is what we're talking about. It's carbon-based molecules. Now, lucky for you, because you're non-bio majors, you probably never have to take organic chemistry, which is great, because it's really hard. Essentially, you have to draw all these little molecules, and sometimes the H is going this way, and sometimes the H is going this way, but it looks exactly the same when you're drawing it on paper. It's kind of crazy. So, like I said, lucky for you, this is about as deep as we're going to go into this organic chemistry right here. So, yay! Now, one thing we are going to learn is that we're talking about macromolecules. So we're talking about these larger molecules, which means they're built from smaller pieces. So the smaller pieces, in fact, an individual piece is known as a monomer. Mono meaning one, right? So this is a single unit. Now, when we talk about each of these four macromolecules, we're going to be talking about different monomers and what makes up polymers. So poly meaning many. So essentially you take a single monomer, right? And you add a bunch of them in a little row, and now you have a polymer, right? A bunch of little single units. Okay, so you will need to know, especially for your exams, you will need to know the difference between each of these different macromolecules that we're going to go into right now, and what their monomers are and what their polymers are. So we're going to talk about that. Don't worry, but definitely write those down when you guys are taking notes today. So the way we make these polymers is through a reaction called a dehydration reaction. Now, dehydration kind of sounds like you're losing water, and essentially that's kind of what's happening. So what you have is this shorter polymer here that you're trying to make into a longer polymer. So what you do is you keep adding those little monomers, 1, 1, 1, 1, 1, and you start making a little chain. Okay, so what you do is sticking off the end of this short little polymer is a hydroxide or an OH molecule. On this monomer that you're trying to attach happens to be an OH, but OHs and OHs don't really like each other because they're also kind of negative, and so they repel each other. They're the same charge, like we learned about last week. They repel each other. But the H is kind of a little positive and the OH is kind of a little negative. So when these two polymer, the short polymer and the short monomer, try to get together, what's going to happen is those two water molecules, which are sharing via covalent bonds, right, these electrons right here, then they're going to actually kick off that water. They're going to kick off the OH and the H, and that's going to release a water. When they release that water, remember, water is a very stable molecule. Water's super happy. Water's stable, it's balanced, sort of, right, because it's polar, but it's stable, and so therefore water goes off on its own. Now we have this open end here for the short polymer, we have this open end here for the monomer. They're going to want to join up, right? Remember, we don't want those loose electrons floating around. You've got a positive, I'm a negative, let's get together kind of thing. Okay, so that's exactly what we see right here. So we have a short polymer and a monomer that we're trying to attach. We kick off the water, hence dehydration reaction. We release a water, and what we get is that longer polymer. Okay, so that's how we make these polymers. In the alternative, in fact, in the reverse, we're going to have what's called a hydrolysis reaction. And that actually takes water or uses water to split up that polymer. So sometimes your body wants to make a polymer. Like when we're making proteins, we want to make a polymer. We have all these little amino acids, which you don't know about yet, but you will. You have all these little amino acids and you're trying to make a protein, right? So that's when you would use a dehydration reaction, right? Because you're actually trying to build something, you're trying to make it longer. Now sometimes your body goes, hey, I have this big glucose or this big starch molecule that I need to chop up. Okay, that would be the hydrolysis reaction. Using water, we're going to break up that starch molecule into little tiny individual monomers, which are glucose. Okay, so sometimes we're building stuff, sometimes we're breaking stuff down. Depending on what we're doing, we're going to use these two different reactions. So building stuff, we're going to be using that dehydration reaction. Breaking stuff down, we're going to be using hydrolysis. And the word lysis actually means to break or to cut. So you can imagine hydrolysis, we're cutting with water. We add water to the polymer, and what happens is, again, that water molecule is going to break up and attach to that shorter polymer and that single monomer. So we went from a larger thing to a smaller thing. Okay, so that would be breaking a polymer chain using that dehydration reaction. Now let's go into the actual 4 macromolecules that you will need to know for your test. You will also need to know the differences between these 2 different reactions, so make sure you guys are paying attention when you're watching these videos and you're taking notes. Alright, so here are our large biological molecules, also known as macromolecules. Remember, a macro means big. Okay, so we've got carbs, lipids, proteins, and what's called nucleic acids. Now, the first couple might actually seem pretty familiar to you, and that's because we use them, in fact, we eat them in our everyday life, and it's really important to maintain a balanced diet because your body needs all of these different macromolecules to make you and to keep you alive and running. So it's really, really important that you have that balanced diet. Even if, say, you are a vegetarian or a vegan, and absolutely nothing against that. I think that's an amazing choice. I'm not so strong, but great for you. But you guys know it's so important to have things like protein in your diet. So that's when you look for like a plant-based protein because you still need proteins. And we're going to talk about why that is in just a couple slides. So let's start with our first macromolecule. Let's start with our carbohydrates. So these are essentially sugars. And if anybody's ever been on a low-carb diet, it's because you're basically told to cut out sugars. And these carbohydrates, or especially these complex carbohydrates, are just built from little tiny sugars. I know, carbohydrates are great, but if you are looking to lose some weight or to slim down or just even kind of, keep a healthy diet, you probably want to keep the carbs to a minimum because we don't need that much sugar in our diet. Although, sugar is an amazing source of energy. So if you've ever heard of like carbo loading, say you're going to go run a marathon the night before, actually, I don't know, I've never run a marathon, but before you run the marathon, what you want to do is you want to eat a lot of carbs, right? You want to carbo load up. You want to just eat all the carbs that you want because in the short term, carbs are amazing for energy. If you're going to have a long day, it's really important to eat some carbs in the morning because those carbs will keep you going throughout the day. However, carbs are a short-term energy source. So after a while, if you haven't run the marathon, say you went out to that Italian dinner and you had pasta and breadsticks and all the carbs in the world, but you didn't run the marathon the next day. What's going to happen is your body's actually going to turn those carbs, those useful little bits of sugar, it's like, well, we don't need it right now, let's go ahead and store it. And unfortunately, you can store it as fat. Yes. So when you are trying to, again, cut out the sugars, or cut out the carbs, it's because really you're trying to reduce the amount of sugar that you're eating. So unfortunately, sugar, breads, all that kind of stuff are delicious, great sources of uses, great short-term sources of energy, but you gotta make sure that you burn off that energy, otherwise it can store in all sorts of bad ways. Now, when we talk about individual monomers, remember the single units, we're gonna be talking about what's called simple sugars. And these simple sugars are simple because there's just 1 or 2 of them, right? A simple sugar would be like a monomer, like glucose, right? Glucose and fructose are 2 simple sugars. There's only 1. Now, when you take a bunch of the glucose molecules and you add them together, and you make that line, you make that big polymer, that's what you're actually going to get what's called a complex carbohydrate, meaning there's lots of different little carbohydrates. There's lots of tiny little sugars to make up a big long chain. That would be something like starches that you would find in like potatoes and breads, right? Which again, if you were to eat those potatoes and breads, essentially you're just eating a lot of sugar. Because when it actually gets broken down, they're just single sugar molecules. They're single, simple sugars. Now, if you were to eat the simple sugars, they're much easier to digest because there's only one of them, right? But something like a starch would have so many more, and therefore you have to break all of those bonds. And remember, when you make a bond or break a bond, it takes energy. And therefore, it's going to take a lot more energy to digest that starch than it would be like a simple sugar, like straight glucose. So, things to keep in mind. Now, when we talk about these simple sugars, we're talking about a monosaccharide. Saccharide means sugar, mono means one. So this would be like our glucose, our very simple sugar. There's only one. Okay, so that would be a very simple one like glucose, which we find in sports drinks and many other different forms of sugar, and fructose, which is found in fruit. Glucose and fructose are essentially the exact same little molecule, except one hydrogen's poking out this way and the other one's kind of poking down that way. So structurally, they're a little bit different, but really they're about the same. And fructose, I think, is a little bit sweeter than glucose, but still basically the same types of molecules. Now, a disaccharide, imagine we had a monosaccharide plus a monosaccharide. That would give us a disaccharide, right? And that would be a hydrolysis reaction or dehydration reaction. If you said dehydration reaction, you would be correct. Remember, we're going from a smaller thing and we're building. So in this case, we would kick off that water and we would make that polymer, right? Well, in this case, it really wouldn't be a polymer because there's not many. There's only two. So a disaccharide is two, right? Two simple sugars. Monosaccharide plus monosaccharide gives us our disaccharide. And again, that would be something very, very simple like lactose or sugar. Sucrose. Lactose or sucrose. And lactose is a molecule that, again, is found in things like dairy and stuff like that, which some people have a very hard time breaking down. And we're going to see why in a little bit. And we're actually going to talk about our good friend lactose and lactose intolerance a couple times throughout the semester to really solidify this information. But we're going to see lactose again soon. So here's what we see. We see some monosaccharides. They all kind of have this circular kind of hexagon shape, just like that. And you can see the glucose, fructose, and galactose all basically look the same, except kind of some of these little molecules that are poking off some of the edges are in different places or slightly different molecules, but really they're basically the same. Now we have sucrose and lactose right here. These are our disaccharides, meaning two simple sugars. So imagine we had glucose and fructose and we just stuck those together. And that's what you'd actually see right here. Glucose and galactose, I believe, is sucrose. No, glucose and fructose is sucrose, and glucose and galactose is lactose. There it is. Organic chemistry coming back to me. It's been a minute. But we see our disaccharides are sucrose and our lactose. And again, you can see one simple sugar, two simple sugars, they're connected, right? One simple sugar, two simple sugars, they're connected to make those disaccharides. Now over here, this is glycogen. This is found in our muscles. This is known as a polysaccharide, meaning the many saccharides or the many sugars. And that's what you can see right here. Again, you see this little circular hexagon shape, and it goes all of these little chains going all around and branching off, and that would be making a complex carbohydrate. So these would be more simple sugars. This would be a complex carbohydrate right here because it's so complex. I mean, look at all those different bonds and stuff. So again, much more complex than the simples. All right, getting to lactose. I told you we'd get there, and again, we're going to get there throughout the semester. So if you are one of my students who are lactose intolerant, one thing I would like to point out is it's not an allergy. So some people don't understand the difference between allergies and intolerance. So if you were to eat, say, or drink dairy or something like that, you wouldn't go into anaphylactic shock and your throat wouldn't start closing up and you wouldn't have the histamines being produced to start swelling up and all that kind of stuff, because that's an allergic reaction. You would actually just have a little bit of digestive issues because what you're lacking is the enzyme that breaks down lactose. And we're going to talk about all about enzymes in just a second. But what you can see right here is, again, this is our good friend glucose, our good friend galactose. We stick them together, we kick off the water, dehydration reaction, right? Release the water. We get two monomers and now we form a disaccharide. So two monosaccharides, now we have our disaccharide and we kick off that water. Excuse me. And again, this would be things in like dairies and cheeses and just delicious stuff. All right, so that was, again, two of the very common disaccharides that we're going to be talking about in the future. So make sure that you guys are paying attention to, again, what the monomers are, what the polymers are, and how these things are actually built via the dehydration reactions. Or if they're breaking them down, the hydrolysis reactions. Now, when it comes to things like these sugars, America loves our sugars. We really, really do. In fact, we eat an astonishing amount of sugar every single year. Up to 100 pounds of sugar per person. Per person. Essentially, you're eating another person built out of sugar every single year, which is just crazy to think about how much sugar that actually is. But there's pretty much sugar in everything. So if you are trying to, lose weight or get healthy or cut out the sugars or something like that, it's actually kind of hard to do because there's so much added sugar in all of our diet, which, could be one of the reasons that America is slightly on the overweight side as an average compared to some of these other countries. But it's really also all the processing that we're doing to our food. We process it so much that it actually doesn't start to break down, and we're going to learn about all that too, which is kind of crazy. So we're going to learn about that shortly. All the processing that is good and maybe not so good. All right, so moving on to our polysaccharides. We have our monosaccharide, there's a single, we have our disaccharide, there's two. Now we have a polysaccharide, there's actually going to be many of them. Okay, so these are known as the complex carbohydrates, like the glycogen we just saw with all the little branches and the little tiny parts. There's so many little parts, hence complex carbohydrate. And essentially they're just built, they're huge polymers of these tiny little monomers, these monosaccharides. So here's a couple of examples, and yes, you will need to know these. Okay, so say we have some glucose and we stick it all in a chain. We build that long chain. That's going to be our starch molecule. That's what you can see right here. This is going to be found in things like potatoes, right? You know that potatoes are pretty starchy. That shouldn't shock you guys. The next one is glycogen. This is going to be found in our muscle tissues. And essentially all you've done is you take a couple chains of the starches and you connect them, right? So you kind of branch them off of each other. So we have one starch chain going here, and then you have a branch here, a branch here. Now it's complicated, and it actually becomes this glycogen that's found in our muscles. Okay? If you are in a plant cell, you have things like cellulose. And cellulose, again, is going to be those long chains of starches, but this time connected this way. So it's actually going to create this like mesh network. And that's really important because starches are always really important. Cellulose is really important for a actual like building blocks, building material for these plants. Remember, we have things like bones and muscles and stuff like that, but plants don't. Plants are essentially just all made out of cellulose. And that gives them that 3D structure, that nice, tough, rigid structure, because they're just long, complicated, complex carbohydrates. Which is why some herbivores can survive just on plants because they're getting tons of energy. Remember, all of this can be broken down into energy. Every time you break that bond, you're releasing that energy. And therefore, these plants actually store a ton of energy inside of them, which is what feeds giant herbivores like elephants and giraffes and these huge bison that just eat tiny little bits of grass. So this is where they're getting all their energy from. Now, in animals, we also use carbohydrates for things like structure. This is a little lobster right here, and he's covered in an exoskeleton made out of what's called chitin. Now, chitin, C-H-I-T-I-N, is actually a complex carbohydrate. So really, that shell is just made out of a bunch of sugars stuck together. So all of these sugars, these complex carbohydrates, can also be used for structure, right? Structure and support. So short-term energy, absolutely. Structure and support, yes, right? As you build up larger and larger molecules, you can actually make things like this giant redwood tree, right? That's all just sugar, which is crazy, right? Just tiny little monomers building these huge polymers, building these massive structures that go 90 feet plus up in the air. It's insane how cool some of this biological stuff is. And hopefully you guys are already finding this a little bit interesting, or hopefully fascinating. All right. Let's get to our next macromolecule, the most diverse of the macromolecules. This is proteins. And proteins are super important to animals, especially important to us. We are like 90% proteins. Like pretty much all of us is just proteins. And every protein is going to be structured a little bit different. And we've already kind of talked about this. Remember, structure leads to function. So if you change the structure, you're going to change the function. And that's true of these proteins. Essentially, at their base level, they're all the same. But it depends on how they get folded and then turned into this structure. And it really depends on their structure, what their function is going to be. So several functions include, we have structural proteins, which again are used for support structure, right? We have storage proteins that are actually going to be storing some of these amino acids, because amino acids are going to be the monomers of our protein. So the protein is the polymer. The amino acids are the tiny, tiny little monomers. Let's see. We have contractile proteins. So in our muscles, we basically have these little rods that go and contract back and forth. And that's actually how we're able to move. So our muscles don't push against anything. Really, all they do is contract and relax. And so these contractile proteins are essentially what allows us to go walking down the street. So thank you, proteins. Transport proteins are also the reason that we are the way we are, especially as large as we are. We have things that's called vessels, like veins and arteries, and it's sending blood all through our body. That blood is carrying oxygen. So it takes the oxygen from my lungs to my little toe. Right? If I didn't have that blood, I wouldn't be able to get the oxygen everywhere it needs to go, and therefore it would restrict the size that I can grow. Luckily for us, we have these things called transport proteins. Transport proteins are essentially our blood. And what are they transporting? Oxygen. Great! So that again allows us to be motile, to move around, to get all the oxygen we need to all of our little cells in our body, and be able to do the things that we need to do every single day. And finally, we have enzymes. Enzymes are really, really crucially helpful for so many reactions in our body. And enzymes are known as a catalyst. And a catalyst speeds up the reaction. So if you're waiting for a reaction, you're waiting. You throw an enzyme at it, boom, the reaction goes forward. This is important because sometimes we have reactions that need to happen very, very quickly, right? And therefore, you can't sit around waiting. You need to send those enzymes in and get that thing moving, right? Get that reaction going. Okay, I'm getting ahead of myself. I was going to talk about lactose again, but not yet. I'm waiting. All right. Did I get everything here? Yes. Okay. Let's see. So remember that monomers of proteins are amino acids, tiny little amino acids. Now, there's about 20 different types of amino acids, and we need basically all of them to survive. Most of them are already in our body and just get recycled all the time, and therefore we don't have to worry about them. But some of them come from our food. And this is where those vegans and vegetarians have to actually pay attention to their diet, because they need to get those proteins, those amino acids, from things like plant-based protein. Because we don't make all of those amino acids, and therefore, to make all the proteins that make us us, we need every single little building block. And if we don't make it, we have to go out and eat it. Okay, so that's where, we eat a cheeseburger, you're going to get all those amino acids that you need from the meat, because it's all protein, right? It's just, you broke down the protein and they're just amino acids. So now you have all those amino acids that you need. If you don't eat things like meat, then you have to look elsewhere for those proteins. And that's where things like nuts and other plant-based proteins are actually really useful for, because you can supplement all those amino acids and be fine. So there's no real reason to eat meat except to get those amino acids. If you can find them elsewhere and you don't want to eat meat, find them elsewhere, and awesome, you're saving animals. If you don't want to go out and eat a bunch of beans, right, then just eat a cheeseburger and you're going to get all those amino acids that are necessary for life. And we're going to see a chart on that in just a little second to see what amino acids you might be missing. If you are one of my vegans or vegetarians, this chart will actually help you to find out how you can get that balanced diet and get all those amino acids that you need. But before we get there, let's talk about each one of these amino acids that I keep bringing up. Now, structurally, they're basically the same at their core. So what we have here is we have this amino group, which is made of a nitrogen and 2 hydrogens. And then we have this carboxyl group, which is made of a carbon, a hydroxide, and a double bond to an oxygen molecule. Now, the only difference is going to be here is what's called this little side group, or this R group. And that can be, many different things. So if we look down here, we can see leucine. And this R group showed here in yellow, or this side group, R group, same kind of thing. Organic chemistry, they call it an R group. This is leucine right here, and you can see that it's got a bunch of these CH molecules. And so that's structurally what makes leucine. And over here we have serine, which only has one CH molecule and then a hydroxide molecule. So again, this little side group can vary, but this base right here is always going to stay the same, okay? Luckily for you guys, again, we don't really talk about the structures and go into this too much. A little bit in the lab, but that's about it. So you don't need to really memorize what's the difference between leucine and serine, and I'm certainly not going to make you memorize any of these amino acids. Just know that amino acids are the monomers for things like proteins, like enzymes and stuff like that. Now, when we talk about proteins, remember I said that they're essentially all just a chain, but they fold kind of different. This is what we're talking about. So when you have the primary structure or the first structure, this is what we're looking at right here. You can see a single amino acid in a chain. Boom, 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 one after the other. And that's what we can see right here. All of these are single amino acids that has now made up this chain. Okay, this would be our primary structure of our protein. Now, as we continue along, and again, this is all through dehydration reactions, right? Kicking off the water, adding the polymer. Kicking off the water, adding the polymer. Okay, so now we take our primary structure, which we can see right here, and now we're actually going to fold it. So again, these amino acids are going to want to kind of form bonds between each other, and that's what they have done right here. These are all covalent bonds formed in between these different amino acids right here. And so these, again, bonds are going to want to kind of form to other bonds and connect and stuff like that. And so you have what's called a secondary structure, which comes in two different forms. You either have this secondary structure, which is pleated sheets, like ladies. If you have a pleated skirt, it's kind of like a folded skirt that opens up a little bit. And then, or you have the alpha helix, which is kind of like a DNA spiral, like that double helix that we're going to be talking about later on in the semester. So these primary structures are connected with covalent bonds, but the secondary structure, these little dotted lines right here, remember dotted lines are always hydrogen bonds. Solid lines are going to be covalent bonds, dotted lines are always going to be hydrogen bonds. So now they're kind of folding and they're like connecting in weird ways with each other via these hydrogen bonds to get this like folding going on. So again, we're going to keep folding. We're going to have a combination of these pleated sheets and these alpha helices, which you can see right here. And that's actually going to make what's called our tertiary structure or our third level structure. Now, if we were to take a couple of these third level structures and stick them together, now we have what's called a quaternary structure or the fourth level. And that's what you can see right here. 1, 2, 3, 4. 4 of these little subunits are actually going to be making up this large protein now. Now, this protein could be anything. It could be a channel protein. It could be a transport protein. It could be an enzyme. It could be lots of different things. And it's all based on how it folds. Now, how it folds is all based on the sequence of amino acids. The sequence of amino acids is based on your DNA. So your DNA essentially gives you instructions on what proteins to make. Is your hair going to be curly? Is it going to be blonde? Are you going to have freckles? All of those are proteins that your body makes to make you look like that. And it's all based on your DNA. And that's what we're going to talk about when we get to transcription and translation, which is later on in the semester. Now, you're thinking, okay, well, there's a lot of these amino acids in that chain. I mean, look how many single amino acids are here. Can you imagine how many are in here or even in here? Hundreds and thousands and sometimes even millions, right? A lot of these amino acids. Well, sometimes things can go wrong. What if you put down the wrong amino acid right there? Won't that change the structure? But the structure determines the function. That's going to change the function, and that's exactly what happens. So here's our good friend sickle cell. And this is an example that we're going to be talking about a lot through the semester, so make sure that you guys really understand what's going on here. So in the top, we have what's called a normal red blood cell. This is what your normal blood looks like, the little hemoglobin that's going to be carrying that oxygen around. Now, it has a very normal sequence, right? It has all these amino acids right here. And when it's given that normal sequence, you get a normal hemoglobin. Now, what happens if just one single amino acid is changed in the whole sequence, right? If it's right here on number 6, and if it happens to go from a glutamamine, I think, yeah. Anyway, if you go from a GLU to a VAL, which is valine, what you get is sickle-cell anemia. So that one single amino acid gets switched to the wrong amino acid, and all of a sudden, instead of folding this way, now you're folding this way. And because you're folding this way, now your red blood cell looks like this. Instead of this big, round, circular, happy red blood cell, now you kind of have this concave, flat, sad-looking sickle-celled red blood cell. And now this doesn't carry oxygen as well as this one. This one is made for carrying oxygen. This one is not made right. It's weird, and therefore it's not going to function properly. And unfortunately, you're going to end up with sickle cell. Now, because if this is a mutation and it actually happens in your genes, that means all of your red blood cells are going to look like this. All of your red blood cells are going to be sickle cell, and you can even pass it on to your kids because it's technically a genetic thing. So not good, but all it takes is one single substitution from the GLU to the VAL, the wrong amino acid, and all of a sudden now you have, and it doesn't really happen like that. Like one day you're not just going to wake up and now you have sickle cell. This does happen like when you're born, usually transferred from parents to offspring and stuff like that, because this is a genetic disorder. So don't worry, you're not going to wake up tomorrow with sickle cell. But yeah, it is a mutation in just one single amino acid. Now, remember, protein shape determines protein function. Protein structure determines function. This is so true. This is going to be something I'm repeating over the semester because it's really, really true. Now, one thing that we would learn in class and lab, but we might not get a chance to, so we might do it online kind of thing, is that there's lots of things that can actually mess up proteins. And this is called denaturing, or to denature a protein. So the nature of a protein is to function a certain way. If you denature, you've stopped it from doing its original purpose. Okay, so if your protein is meant to work, right, denaturing means your protein is not going to work anymore. It's not going to function anymore. And usually what happens is you're messing up the folding. So remember the folding and the bonding, and it kind of goes like this with the hydrogen bonds? Well, now you start to kind of break the hydrogen bonds and your protein just kind of falls apart. And there's lots of different things that can actually make this happen. One of them is temperature, right? You know that if you get a temperature above 104, you need to go to the hospital because at about 105, 106 is when your proteins start to break down. Okay, so essentially your whole body would just stop working. Remember, if we're mostly proteins and all of a sudden that mostly protein stops working, you're not going to be able to function and you're probably not going to be able to survive. So temperature really, really affects proteins, which is why we maintain a set temperature level. Right, we are that 98.7. We stay right there. We don't go too high. We don't go too low. Very, very important to maintain that temperature because it affects our proteins. What else? Oh, pH. pH is another one. So we're going to learn about acids and bases and stuff like that. You really want to keep your blood pretty, just slightly on the basic level. You don't want to get it acidic. You don't want it to get too basic. You don't want it to go either one way or the other because it can really affect it and mess it up. So again, all of these proteins are really, really fragile. And so things like temperature and pH can absolutely affect them. So things that, your body regulates on a regular basis. All right, so let's talk about our good friend lactose and lactase again. So remember that enzymes are proteins. So enzyme, again, is going to be one of those polymers of those amino acids. So enzymes are used to speed up reactions. So what we have here is our lactose molecule. Our lactose molecule, again, that's going to be found in the dairy and the cheeses and stuff like that. Now, we have this lactase enzyme, okay, that's going to break down lactose. So anything that ends in ASE is usually chopping something up or breaking something down. So lactase specifically is meant to break down lactose. Okay, that's why it's named like that, because it only has one function, to break down lactose. Now, what this is called is what's called a lock and key model, meaning the lactose will fit perfectly inside the lactase. Okay, so the lactose would be the key and the lactase would be the lock. Now, there's only one key for one lock, and that's kind of the same thing for enzymes. They're very what's called substrate specific. Now, substrate is the lactose. The substrate just means substance. It's a fancy word for substance. So don't let that word kind of confuse you and be like, oh, I don't know what a substrate is. It just means substance. So in this case, excuse me, gross. In this case, each enzyme is specific to the substance. Okay, so the lactase, again, is going to be used to break down lactose. Okay, you can't break down anything else. It only can break down lactose because lactose is the only thing that actually fits inside lactase. So this is our enzyme, and this is what's known as the active site. The active site is where the enzyme gets activated once something is there. Okay, so the lactose right here is going to fit perfectly inside the lactase, and that's going to activate the enzyme. Now lactase is like, oh, I have to go do my job. My job is to break this bond right here. Okay, so I need to break down these sugar molecules. I need to break that bond. Okay, so that's its goal, and that's what we're going to see right here. So we have our lactase is the enzyme, right? The lactose is the substrate. The lactose is sitting in the active site of the enzyme. Now the enzyme is activated. It goes, cool, I'm going to do my job. It starts to break that bond right there. Now it releases glucose and galactose, right? It's broken the bond between them. Now they're not a disaccharide anymore. Now they're monosaccharides. Right now we're trying to break stuff up. Okay? Now if you are lactose intolerant, again, it's not that you don't have the enzyme, but your enzyme is a little misshapen. So all it takes is this little structural change right here, and now this lactose molecule won't fit inside the enzyme. If it can't fit inside the enzyme, then it can't be catalyzed. That bond is not going to be broken. So now your body has to actively sit there and put all this energy in to breaking this little bond right here. Whereas normally the enzyme would just do it like that, right? It catalyzes your reaction, speeds it up. But your body doesn't have that ability because the lactase enzyme is a little bit misshapen. Structure leads to function. You mess up the structure, you mess up the function. Now your body has to deal with lactose on its own. Okay, so that's why you're not allergic to it. You don't die from it. You just, it's really hard on your body. It's kind of hard on your digestive system because you don't have the ability of this enzyme to break it down. Now, here's that chart that I promised you guys if you are my vegans and vegetarians and you are looking for some alternative amino acid sources. So what you can see right here is these are all the amino acids that you absolutely need to ingest. And so here's some of the places you can look for it. Okay, if you are eating things like milk, tofu, chicken, or a T-bone steak, notice all of these amino acids are present. And that's again because these are animal-based, they're just full and chock full of all of these proteins and therefore these amino acids. So if you're looking to substitute, right, if you were to look at, say, something like white rice or lentils, they both have all but just one amino acid. So really, and it's not even the same amino acid, right? So white rice is missing lysine, but lentils have lysine. So all you need to do is eat white rice and lysine, or white rice and lentils, and you get all your amino acids. So this is a really helpful chart if you guys are looking for some alternative sources to meat-based protein. All right, getting on to our next group of macromolecules, we are talking about the lipids, also known as the fats. If you hear me say lipids and fats, it's kind of interchangeable, and that's because they kind of are interchangeable. Notice this little guy right here, right? This little fat. Oh, isn't he cute? This is a seal. And he is covered in lipids, right? In those fats. And for him, fats are a really, really great source of insulation. So he basically pats his body with this layer of fat and that keeps the cold out and the warm in. So he's able to survive in these cold Arctic, habitats. Lipids are also really great for energy. Now, remember we talked about carbo loading and then you didn't run the marathon and it all got turned into fat? Well, that fat is still all that usable energy. You could absolutely break down that fat and get a ton of energy. And that's kind of what weight loss is. You're trying to burn off more fats than you're bringing in, and therefore you're going to start to burn off the storage that you have. Great for long-term energy too, as long as you're breaking it down. If you don't break it down, it's just going to keep accumulating and accumulating. But really, really great for things like cushioning, insulation, and all sorts of other great things. And totally necessary. So you can never just cut out all fats in your diet. You still need fats, but what's called like the good fats kind of thing. Now, this is a monomer right here. It's a type of monomer that we have. We have two different types. We have the triglycerides and the phospholipids. Those are both monomers. This right here is called a triglyceride because you can see 1, 2, 3 tails. Okay, so this is known as the glycerol head right here, and then you have your fatty acid tails coming off here, hence triglycerides. That's 3 tails coming off of that. Now, again, this is not organic chemistry. I'm not going to have you draw these, but I do want you to at least recognize each one of these macromolecules and their structures. Now, there are 2 different types of fats. Remember, we talked about the good fats and then the bad fats. The bad fats, I hate to say it, are all probably the delicious ones that you like. So what we can see right here is these are the saturated fats, and they're all called saturated because they're covered in these hydrogen bonds. Now, they all have a single bond from a carbon to a carbon to a carbon to a carbon, and hydrogens off either side of the carbons. Now, that keeps the carbon molecules very happy because they all have that octet rule. Remember the rule of 8 that we talked about? But that means that their tails are also going to be very, very straight because it's single bond to single bond to single bond to single bond. So they have very, very straight tails. This is why they're called saturated, because when you stack them next to each other, you can really pack them in. You can really saturate them in small areas. So now you're getting a lot of these fat molecules in a very small amount of area. That would be things like the cheeses, the eggs, basically anything probably delicious that you're going to like. Now, these guys are typically solid at room temperature. Okay, so think of like a cheese. It's usually solid at room temperature. And again, that's because you've packed them tightly in together. So they're really, really solid. They're not going to move at all. They have no room to move. They're just packed in. That is different than the unsaturated fats that we can see right here, which are typically going to be liquid at room temperature. And that's because they have these kinky little double bonds. And I don't mean kinky like kinky, but I mean literally they're kinked. So they kind of go this way, and then this way, and then this way, and then this way, and then they just kink back and forth. Because of their kinky little tails, they don't saturate and they don't get as close together. So in the same amount of space, you actually have less molecules because they're so spread out. So then you have less fat in the same area, and therefore it's less dense, and that's the kind of liquidy that we get. But these are the good ones. These are the ones that are easier to break down. These are the ones that are a little bit better for you. These are known as the good fats. So they're not saturated with hydrogen. They're not saturated together. They're just, the fats. Now, the hydrogenated fats, these are the ones you really have to worry about. This is the problem that we have here in America is we love to process our food and we love it to last forever. I want to make that Twinkie sit on the shelf for 3 years and not go anywhere. Well, no bread anywhere should last 3 years, but this is how it happens, is they do what's called hydrogenation. And essentially you're taking those kinky little tails and you're artificially straightening them. So when you do this, now you can pack more into the same area, but you've also kind of chemically messed with it. So now it makes it really difficult to digest. In fact, really difficult to break down. That's why it lasts on the shelf for years. So it's great for things like flavor and mouthfeel and texture and shelf life, but it's not so good when it's sitting in you, not going anywhere because your body's not able to digest it. That's the whole point of this hydrogenation. It's so that it can't break down, so that it can have a better shelf life. But your body is supposed to break down food, so not so good for you. And again, a lot of American companies, unfortunately, are doing this. We love them. Tastes great. I'll be the first one to admit that I totally eat some. But it's something to keep in mind that you would, maybe have to eat a little less of, or just every once in a while kind of thing, like as a treat. I don't know. Here, getting to the other type of monomer that we have, phospholipids. And we are going to see phospholipids numerous times in the semester, so make sure that you guys are paying attention to this. Right, so we have a little phosphate head right here, and then we have our fatty acid tails coming down here. Now, that little phospholipid head is what's known as hydrophilic, which means it really likes water. So it can be around water, which is great because these phospholipids actually make up our membranes. So you can think of a membrane as kind of like a protective layer, right? You don't want things just randomly floating into your skin. You don't want things randomly floating out of your skin. You need to keep things where they are and keep things nice and protected. That's kind of like what a membrane does. So because the head actually likes water, it can be on the inside of the cell and it can be on the outside of the cell because there's water in both places. But the tails that we can see right here, which hang down, and we can also see right here on the inside, are hydrophilic, meaning they don't like water. They're scared of water. They're like, get away from me, water. So if you were to take up a bunch of phospholipids and shake them and pour them into a jar, what they would do is they would orient themselves like this, with the hydrophilic heads on the outside touching the water, because they like water, and the other way around. Hydrophilic, meaning they like water, heads on the outside, hydrophobic, tails on the inside. So the tails are like, oh, we're all going to hide here, and we don't like the water, and the heads are on the outside going, I like water, this is okay. So this is what actually makes that membrane so that things can't just pass in and out of your cells. All of your cells, in fact, all cells have this phospholipid membrane. All cells everywhere, plant cells, bacteria cells, they all have this membrane. Okay, so it's really important for keeping things to go in and out of your cells. It's maintaining like a barrier so that you don't just have, your mitochondria wander out of your cell. You kind of need that. So important to have. So every membrane in your body is going to be made out of this phospholipid membrane, which is great. And we're going to see it again in the semester a couple different times. So always made out of these phospholipids. Great for membranes. Another super important type of lipid. Lipids, you're like, wait, fats are so important? Yes, they are. Hormones, all of your hormones, including your growth hormones, your sex hormones, all of your hormones, all come from cholesterol. Cholesterol is known as the base hormone, and it is a fat. It is a lipid. So you need cholesterol. You need to have all of this kind of fats coming in. However, you don't need too much of it. So when you get too much of it and basically your cholesterol level gets too high, that's where you get things like artery blockage. So you have your vein or your artery, and then the goop starts building up, and then your blood can't, and that's when bad things can start happening. So you don't want that to happen, but you do need cholesterol. Cholesterol is a base hormone for things like estrogen, testosterone. These are all really important hormones that we need to have all of our body functioning the way it does. So this is why you can't completely avoid fats. You do need the fats. Luckily, there's fats in almost everything, so you're probably going to be fine. But yeah, steroids and hormones, very, very, very important, and a type of lipid. Waxes. Waxes are another type of lipid. Waxes are super important in things like plants. Plants have what's called a waxy cuticle, and the cuticle essentially is a nice waxy layer, and that's why water runs off plants, beep, right off. And that's actually to help prevent dehydration. So plants can't go take a sip of water whenever they want to, and therefore they need to keep and maintain whatever water they have. They need to make sure that nothing gets evaporated out of them. So they have that waxy cuticle to protect the outside. We also have wax, right, in your ear. Why do you have wax in your ear? It's actually to keep foreign antibodies from floating into your body. Yeah. We're going to learn all about microbes and the stuff that's floating around in the air, and you are going to thank your immune system that this is actually keeping you healthy and alive. Because even the wax in your ear, which is like, ew, gross, no, it keeps little things from floating and getting into your brain, which is crazy to think about. And finally, we are on our very last macromolecule, our nucleic acids. Arguably, maybe the most important one, because, we wouldn't definitely be here without these, but then again, you really wouldn't be here without all of the macromolecules, so you really can't say which one's more important. But we can say that the nucleic acid is all of our genetic material. So what makes you you? Remember those instructions to your proteins, right? These are nucleic acids. These are your instructions for everything. Okay, so the reason your hair is curly is because of your DNA, right? It sends messages to your ribosomes. Your ribosomes make your proteins. Your proteins come out a certain way based on the instructions that the DNA sent. Okay? So all your genetic information comes from these nucleic acids. Now, there are two types of nucleic acids. There's the deoxyribonucleic acid, that is your DNA, right? And then there's your ribonucleic acid, which is your RNA. Okay? Both are types of genetic information. Basically, your DNA is kind of like your master blueprints, and your RNA is like Xerox copies of the master blueprints. Still very important, right? Especially if you're building a building, you can't just have that one copy. Right? You want to keep that one copy locked in the office, and then you just want to Xerox it to all the contractors. That's your RNA. But we're going to talk about all that coming up. So this is what happens. This is known as the central dogma of biology. It goes from DNA to RNA to proteins. Now, your DNA doesn't turn magically into RNA, and your RNA doesn't magically turn into a protein. They're just instructions. Okay, so the instructions get copied down, and the copy is the RNA. The RNA then goes to the ribosome. The ribosome grabs a bunch of, well, we're going to learn about that. I don't want to say that because it's oversimplifying it. But essentially, your ribosome is going to help get all these amino acids and put them in a chain and make your protein. Okay, so it doesn't magically turn DNA into RNA to protein, but the DNA makes a copy of itself. The copy is the RNA. The RNA takes goes to the ribosome, and the ribosome actually assembles a protein out of your amino acids. So this is what's going on here. And we're going to learn all about this again in transcription and translation. All right, so our nucleic acids are built out of these little nucleotides. So the nucleic acid is the polymer, the nucleotide is the monomer, okay? So each nucleotide is built out of a couple different parts. So one, we have what's called a sugar phosphate backbone. Okay, so we have our little sugar right here, kind of looks like glucose, right? And we have our little phosphate group. Okay, this is the backbone. Think of our twisting DNA as a ladder. Okay, so the rungs right here, sorry, I guess the sides? I don't know, ladder parts. These sides right here would be the sugar phosphate backbone. Now the rungs that are going up here, coming across here, these are known as the nitrogen-containing bases, and they're all slightly different. In fact, there's only 4 of them, A, T, C, G. adenine, thymine, guanine, and cytosine. And again, they're going to pair up, and that's what's going to create the rungs. So you're going to have one over here and one over here, and that's going to make your rung of the ladder. And then this would be your sugar phosphate backbone. And then you go, and that's going to be your actual nucleic acid. So we have adenine, thymine, guanine, and cytosine. These are the four that you can see right here, and this is how they pair up. So this is called Chargaff's rule. We're going to see this in a slide or two, but I'm going to let you know now. A always goes with T, C always goes with G. When you're talking about DNA. If you're talking about RNA, A always goes with U, C always goes with G. Okay, so there is a big difference between RNA and DNA, and yes, you will need to know it. In fact, this is a couple of the differences right here. One, we have the U instead of the T. So instead of the thymine that we normally have in DNA, you just replace any T with a U. Okay, so we don't have thymine, we have uracil when we're talking about RNA. So I'm going to give you guys a quick example right here of if we were doing this going from DNA to DNA. So remember, A goes with T, C goes with G. So here's my example. Okay, I have DNA, here's my given DNA strand, right? T, A, C, G, A. If I was going from DNA to DNA, right, there's a T in DNA. So I would just go from A to T to C to G. Okay, so T would become an A would become a T, C would become a G, G would become a C, and A would become a T. Okay, so this is how I would do if I was given this strand. This would be my what's called complementary strand, right? A can only ever go with T. C can only ever go with G, and that's what we can see right here. Look, A only fits with T. C only fits with G. It can't fit any other way. So this is if we were copying one strand of DNA to the other, this is how we would do it. Now, when we're talking about RNA, we don't have that thymine, we have the uracil. So in this case, what we would do is we're going from DNA to RNA, right? We would start off with the same strand we did. We'd have the TACGA. But now we go from always still go from T to an A. This is where people always mess up. I will tell you this on the test. People automatically just kick out all the A's and just go everything U. So they go U, G, C, U. No. The A is still there. So what I always tell people is to write it out like DNA first, but then take every T and just turn it into a U. See? Just every T and turn it into a U. So T goes with A goes with T normally, but we're talking about RNA here, so A would go with U. C would still go with G, G would still go with C, and A again goes to U because there's no T. Okay, because this is talking about RNA. So there is no uracil, there is no thymine, there's only uracil. Okay, so this is how you know if you're talking about DNA or RNA. Is there a U? There's a U, it's RNA. Another difference is RNA is single-stranded. So this would be DNA right here. This would be well, there's a T here, so that wouldn't be RNA. But imagine this is a U here. This would be RNA. It's single-stranded. It's not the double helix. It's just a single strand. And finally, RNA is a ribose sugar instead of the deoxyribose. So it's just a different sugar phosphate background. Remember, this is the sugar phosphate background. It's just a different sugar, that's all. But I would know the difference between RNA and DNA and make sure that you guys can at least sum up this chart right here. Now, again, this is Chargaff's rule. This is, again, A always pairs with T, C always pairs with G if you're talking about DNA. If you're talking about RNA, A always pairs with U, and vice versa. U always pairs with A, and G always pairs with C. I can't talk today. Okay, but you can see that clearly right here. And that's the example that I showed you. And yes, you will have to code this, so make sure you practice. Remember how I said everybody always gets it wrong when they go to RNA. They always just add a bunch of U's, too many U's. Okay, so just go do this one first. And then if it's RNA, just change every T to a U. Don't change the A to U's, just change the T to a U. All right, so here is a chart that is summing up all of these macromolecules that we just learned about, the carbs, the lipids, the proteins, and the nucleic acid. Here's a picture of all the structures on what they look like, how they're different, and some of the functions. Yes, you need to know the functions. Yes, you need to know examples. Yes, you need to know monomers. And yes, you need to know the differences between, some of these functions and stuff like that. So, with that, hopefully you guys enjoyed this lecture. Thank you so much for sticking with me. Remember all those hydrogen bonds. And I will see you guys next week for Chapter 4.